So, we are starting with our next session. A very warm good afternoon to all of you. Nikita, on behalf of ICBILS, will be presenting this session, that is oral presentation, which include Young Promising Research Award. The judges for the session are Dr. Smita Rastogi Verma ma'am, Dr. Kriti Bhandari ma'am, Dr. Navneeta Bharadwaj ma'am, Dr. Prakash Chandra sir, Dr. Simran Bhullar ma'am, and last but not least, Dr. Rashmi Kataria ma'am. Guidelines for this session are each participant will be given 5 to 7 minutes for presenting their work, and additional 2 to 3 minutes will be for a question and answer session by judges. Two presentations will bag titles of Young Promising Research Awards along with a cash prize of INR 2500 each. The selection of award winning participant will be upon on the judge's decision and it will be final. So we are beginning with our session. Our first candidate for oral presentation session is Khushbu Islam.
Hello, should I start?
Yes, you can.
Okay. Good afternoon, everyone. I will be going to present on the topic study of the role of R2R3 MIP transcription factor genes in pungency development in capsicum species. So capsicum is a unique member of Solanaceae with its characteristic property of pungent flavor, which is due to alkaloids, mainly capsaicin and dihydrocapsaicin. But these pungency levels are not same in all the genotypes in capsicum. It is highest in chinense, moderate level in frutescens, and low pungent in anum. Capsaicin and dihydrocapsaicin are important alkaloids. They are reported to have antioxidant properties, anti-cancerous properties. Also, they are used as medicinal as medicines in arthritis, pain, and fever. Therefore, their economic value is high, and we need capsicum varieties with high pungent levels. Recently, there have been reports of the involvement of MIB transcription factors in the regulation of capsaicinoid biosynthetic pathway at the transcriptional level. MIB 31, 108, and 48 are already reported. I have here identified the MIB transcription factors at the genome level in three capsicum genotypes: Annum, Chinense, and Bacatum. First of all, in the identification pipeline, I had performed BLAST-P analysis using the full-length protein sequences of the three capsicum genomes. And I had used Arabidopsis and tomato R2, R3 MIP transcription factor protein sequences as query. In the BLAST outputs, I performed a domain identification using PFAM database, CDD, and SMART database. And finally, I got 164, 159, and 149 MIP transcription factor genes in Annum, Chinense, and Bacatum genomes. After the identification, I performed the characterization, in which, first of all, I mapped the gene positions of all the MIP genes on the 12 capsicum chromosomes, and also I co-localized the previously identified QTLs, which are responsible for the capsaicinoid content, along with the MIP genes. So through this analysis, I had found at least three MIP genes which are lying inside the QTLs on the capsicum chromosome. As we can see on chromosome number 2, we have MIP73, which is lying inside two QTLs. One is responsible for the total capsaicinoid content, and the other is responsible for the dicapsaicinoid content. The other two MIP genes were 48 and 83, which were also lying inside a QTL, which was responsible for the total capsaicinoid content in capsicum genotypes. So this study will help to select the candidate genes for further expression and validation studies to prove their involvement in the capsaicinoid regulation. So further, we carried out the transcriptomic sequencing and analysis to find the abundance of transcripts and the expression levels of all the MIP genes in 3 capsicum genotypes. We selected the fruit stages because the fruit has the pungency in the pericarp and placenta and not the vegetative tissues. So therefore, we selected 2 fruit stages from each differentially pungent genotype, Annam being low pungent, Chinense being high pungent, while frutescens being moderately pungent. Using this analysis, we selected 27 differentially expressed MIP genes for the QRT-PCR validation. In the QRT-PCR validation, we had validated 15 out of 27 differentially expressed MIP genes, and individually, as we can see in Annam, we had validated 8 MIP genes to be showing similar expression trends in the QRT-PCR and the RNA-seq. And in Chinese, we had found 11 MIP genes showing similar expression trends in QRT and RNA-seq, and the corresponding number in frutescens was 10. Now, out of these 15 MIP genes which were validated for their expression levels using QRT-PCR, we found that 5 MIP genes were showing significantly higher expression in the pungent capsicum genotypes as compared to the low pungent capsicum genotype. As we can see, MIP19 expression in the fruit stage at early fruit stage is increasing from the low pungent genotype to moderately pungent genotype to the high pungent genotype. Also, MIP94 was showing significantly high expression in the pungent genotype as compared to the low pungency genotype. MIP91 was also showing the expression levels which were high in the early and the breaker stage as compared to the low pungency genotype. MIP31 and 48 were already reported, but we have validated and verified their expression levels here also, and we can see that 31 and 48 are showing higher expression in their fruit stages in chinense and frutescens, which are pungent, as compared to the low pungency genotype Annam.
Please try to
Now,
wrap in next 2 minutes.
Okay. So there are after the following the expression analysis, we looked out for the sequence variations. We found 129 base pair deletion in the MIP19 in Annam and a 23 base pair deletion in MIP94 in Annam. 23 base pair deletion in MIP94 was caused by a pre-occurrence of a stop codon in Annam sequence, while it was 23 base pair after in Chinese sequence. As it is visible with the difference in the intron length in Annam and Chinese in between the exon. It is 820 here in Annam, and in Chinese it is 797 base pair due to the 23 base pair deletion which went into the intronic region in Annam. So using this study, I had identified candidate MIP genes which will be further investigated and validated using silencing and overexpression studies for their role in the capsaicinoid biosynthetic pathway. I would like to thank my lab members, especially Abdul Rauf sir, my supervisor, and my staff members in SLS, faculty in JNU, and the organizing committee for selecting my presentation. Thank you.
It was nice having you. Any questions from judges?
Khushbu, which tool did you use for co-localization studies?
A map chart.
Map chart. So was it Synteny based?
No, Synteny, it was not Synteny based, it is just the absolute gene locations. Syntenic study I also carried out, but I had not shown here due to limitation of time. But I had carried out the Syntenic study to identify the Syntenic MIP genes also.
Okay, fine. And at what stage was capsicum when you performed all these studies? What is the duration for which you grew the capsicum?
We had sown the seeds and grown the plants until the maturity at the fruit stage. So we collected the early and the breaker stages, which is 5 to 10 days post anthesis, and breaker stage is 35 to 40 days post anthesis. And also the leaf tissue was collected.
Okay, fine.
Thank you.
Okay, thank you so much, Khushbu. So our next participant is Fariba Bushra.
Fariha Bushra.
Sorry, Fariha Bushra.
Am I audible?
Yes, you are.
Is my screen visible?
Yes, it is.
Let me just get into the full screen first. Okay, shall I start?
Yes,
Shall
you
I
may
start?
now.
Okay. Hello, everyone. A very good day to all of you present here. I'm Fariha Bushra from Bangladesh. I completed my Bachelor of Pharmacy from BRAC University, currently working as a research assistant at Drug Delivery and Therapeutics. The name of the topic that I'm here to present today is delivery of monoclonal antibody using 3D printed hollow microneedle. Here's a list of the topics that I'm here to cover. This study has been conducted with the objectives of introducing novel 3D printed hollow microneedles for the delivery of a monoclonal antibody and also to investigate the release profile of hollow microneedle mediated delivery of denosumab in vivo. From the overview of this research work, it's pretty evident that we have employed 3D printed hollow microneedles for conducting the in vivo animal study by ensuring the delivery of this monoclonal antibody. In today's world, with the recent advancement of science, three-dimensional printing technology has made it possible to fabricate diverse microneedles depending on the purpose of application. That has allowed us to upgrade its resolution and integrity, to improve the biocompatibility issues, and also to facilitate the integration of microneedle in different devices that has brought tremendous development in the field of transdermal drug delivery. So before explaining why did we choose to introduce this particular monoclonal antibody, let me familiarize you all with a quick fact. Each year, about 8.9 million people are being affected with a skeletal system disorder called osteoporosis. And if we can manage to establish a successful release profile of this monoclonal antibody, then we would be able to optimize the health outcome of a large segment of people, especially prevailing among the postmenopausal women. So it's basically a Ig2 human monoclonal antibody that works by preventing the interaction between rankle and rank that inhibits the formation of osteoclasts, which are the cells responsible for bone deterioration. Even though subcutaneous injection treatment is available, but often due to trypanophobia, which is the fear of needles, a large segment of people fail to adhere to the treatment plan, which hinders the expected outcome of the treatment strategy. So we decided to opt for hollow microneedle to deliver it because of its nature of being non-invasive, because of its advantage of dose adjustment. As it is single administration, it's less complicated, it doesn't require external assistance, and improvement in patient compliance is also observed. From the picture, it's evident that the microneedle doesn't involve the pain receptors, so it doesn't induce any pain. An optical coherence tomography was also conducted as a part for the assessment of width and penetration of this microneedle. And in the experimental design, it was found to be 11% in case of control group, and 3D hollow microneedle. In case of 3D hollow microneedle, it was found to be 79%. For this study, the materials involved the use of Denosys 60 from Inceptive Pharmaceuticals Limited. 3D printed hollow microneedles along with some reagents and consumables were also included. Prior to 7 days before initiating the experiment, an animal model was selected and isolated, and at the end of each week, an estimation of the plasma level measurement of this antibody was taken to monitor the performance in 3 of the groups of this animal model. The group 1 was untreated, group 2 was treated with subcutaneous injection, and group 3 was treated with hollow microneedles. Each group consisted of 6 mice. The needles were inserted into the dorsal skin, followed by removal after 5 minutes. And to prevent the dislodgement, the patches were also secured with the help of adhesive tape. The trial was conducted for 1 month, and the result was accommodated for a period of 28 days. The result was monitored in each of the groups, and when the results were manifested through the graph, it exhibited that it was successful in mimicking the release pattern obtained usually through subcutaneous injection. So we can see that the successful delivery is possible through maintenance of steady state concentration, which ensures improvement in patient compliance. During the treatment procedure, another benefit was also observed.
Faya, 2 more minutes left.
Okay. During the treatment procedure, it was also observed no penetration mark was left within a span of 5 minutes, which works to the advantage of the patient. In conclusion, we can say that the successful delivery of this monoclonal antibody makes it an alternative choice to the conventional drug delivery tool, ensuring maintenance of steady-state concentration. So exploration of this in future will open new treatment options. Incorporation of MEMS will help in designing personalized medicine. Increase in the permission of drug will help in integrating micropumps and electroporation. Improvement in patient compliance can also be observed in patients with osteoporosis, not only in women, but also men receiving androgen deprivation therapy. Here is a list of references. I'd also like to acknowledge the contribution of my honorable supervisor, Dr. Mohammad Joshimuddin sir, from the university, from BRAC University, and also my Professor Dennis Doromis from the University of Greenwich for the generous funding on this project. They have been there to guide me through each stage of the project. Thank you everyone for listening. If you have any questions, feel free to ask.
Any questions from judges?
Okay, Fariha, I have a question. Can you hear me?
Yes, sure, sir.
Awesome. Okay. I have a question like generally what happens like when we use such kind of microneedle, they compromise with the half-life of this kind of MABs and all. What are your approaches to make it comfortable for the uniform delivery?
Okay, thank you, sir, for your question. If I did get your question, are you talking about how can we prolong the half-life, or how can we ensure that the
Actually, I
drug
have
is...
two questions.
Okay.
Yeah. Both I have two questions. What first one is the how can you improve the half-life, and second one is how can you enhance the uniform delivery, because generally in case of microneedles that complete delivery sometimes lacks. So what are your approaches basically?
Okay, so thank you, sir, for your question. Well, this was also a question that I have encountered from my personal curiosity. So basically, when as this monoclonal antibody is delivered, we have seen that it was quite successful in maintaining a steady state concentration. But to ensure that the half-life is maintained, we need to carry this study for further period. And due to time constraint, and as it is still an ongoing project, so this is something we're trying to keep it in mind. We will, but incorporation of anything like electroporation or incorporation of ultrasound can help in improving the permeation, prolonging the half-life. So that's a recommendation that we will be working on in the near time, because it's an ongoing project, but it is a work in progress actually. So we need to work more to find it out. We have something in mind, but we have to incorporate this ultrasound or micropumps like this. I hope it helps.
Thank you.
Thank you so much.
Okay, thank you so much, Pareha. Our next participant is Madhulika Singh.
Please enable me to share the screen.
Yeah, please.
Yeah, good afternoon to all of you. Myself, Madhulika Singh, PhD research scholar in DTU. My topic of research is elevation of salinity stress by P. indica and plant growth promoting bacteria in HD 2967 wheat variety. Agriculture is the backbone of India, providing food security and economic growth. India faces a risk of decline in growth of crop yield, and for this increasing population, environmentally unsustainable farm practices and environmental stresses are to be blamed. Stress are of two types, abiotic and biotic. Biotic is imposed by organisms, fungi, bacteria, and environmental stresses are cold stress, salinity stress, etc. And my area of research is the salinity. The causes of salinity may be the natural source, artificial source, like irrigation, fertilizers, and the effects of salinity on plants are water deficiency, nutrient deficiency, etc. And effect on soil health are reduced soil fertility, soil texture is also affected, and reduced water holding capacity. Now, the world population will be around 8 billion in 2020, and it will be a challenge to feed all of them. To feed this growing population, the world needs to increase agriculture productivity, efficient resource management, and crop improvement for evolving better breeds and and can help to overcome these stresses. Intensive. There is a need to develop simple, low-cost biological methods in sustainable and environmentally friendly manner without the use of chemical fertilizer to combat abiotic stresses. For that, microbes could play a significant role. And these are the following roles of the microbes. That is, they help in osmolyte production, nutrient uptake, phytohormone production, removal of ROS, soil aggregation, and also in ion regulation. Now, these are my following objectives of the research. Now, these are the beneficial roles of the soil microbes in combating abiotic stresses. This is a review literature, but in all these study, the role of P. indica, glomus, enterobacter in alleviating salt stress in HD2967 wheat variety has not been studied. In my experiment, I have taken the seeds and the seeds were sown in the pots, and these are the following. Treatments which I have given, and the fungi and bacterial inoculum were inoculated in these pots. And after 75 days, these are the pot experiment, control experiment, and the second one with the bacterial inoculum. And these were the following salinity treatment which I have given. The number one is the control, and the salinity levels were from 0, 50, 100, and 201. And in the experiment, the fungi were inoculated, and in the next experimental, the bacterial consortium was inoculated. And then after 75 days, the crop were harvested, and these are the following parameters which I have studied: growth parameters, physiological parameter, biochemical, and the nutrient parameters. Now, I have found that in the microbial inoculated plants, the level of the protein, the level of the proline, and the total sugar at 200 millimolar NaCl concentration was high as compared to the control one. And also I have studied the physiological parameters like photosynthesis, transpiration, internal CO2, and stomatal conductance. And here also, again, at 200 millimolar NaCl concentration, the microbes were able to enhance the photosynthetic rate as compared to the photosynthesis, as compared to the control one. And the same were observed for the transpiration and for the stomatal conduction and internal CO2. And it was found that here the leaf area was also increased in the microbial inoculated plants at 200 millimolar NaCl concentration. The next one, the growth parameters were also studied, like the shoot biomass, root biomass, shoot length, and the root length. And here also you can see that at so much high concentration of NaCl, the microbial inoculated plants show a better shoot biomass as compared to the control. Same with the root biomass, and the shoot length was also increased here. You can see in both the microbial inoculated plants, and same with the root length. Next one, the chlorophyll A, B, total chlorophyll, and carotenoids were also analyzed in HD2967 wheat variety. And here also you can see that the chlorophyll A level was so much elevated here in the microbial inoculated plant at 200 millimolar NaCl. The total chlorophyll was also enhanced, and the carotenoid. I think this is a big achievement here because these microbes, they helped in the growth of the HD2967 wheat variety. And also the membrane stability index also, that is the membrane stability index. The structure of the membrane was also stable here at 200 millimolar NaCl in the microbial inoculated plants. Now, the significance of my work is the microbes, they act as a biocontrol agent, improve soil quality, enhance crop growth and productivity, create a more sustainable agriculture, and they are environmental friendly. Thank you so much.
Thank you so much for presenting. Any questions from judges?
Madhulika, what is the effect of salinity stress on the grain development? You are talking about the area, the root length, and the shoot length, but ultimately in wheat, the part of economic use is the grains that we use.
Yes,
so what is the effect of your the salinity development of grains, the overall productivity, you can say?
Yes, thank you so much for your question, ma'am. Actually, in the last season, when I have grown this plant, I have harvested till the leaf stage. That is after 75 days. I have mentioned in my slide that I have harvested the plant after 75 days, only to study the leaf roots, physical and biological parameters. But now again, I have grown the wheat plant, and now I'm going to analyze the grain biochemical analysis. I'm going to do now. And because of the lockdown, I harvested the plant earlier. And now, again, I am growing the plant and I will do the analysis of the grain now. Because you can see here that there are so many treatments I have made. So studying too many parameters difficult for me in one go. So half analysis I have done right now. And again, I'm doing the plant and I will study the grain analysis again. And the plants are now, I think, 1 month old.
That I understand that you have not performed that experiment until now because of all the pandemic conditions. But can you comment on?
Yes, ma'am, definitely. Yeah, sure, ma'am. Because
what's in the yield due to salinity?
Yes, ma'am. Actually, when these microbes, they are helping in the shoot root growth and they are helping in elevating the biochemical parameters like sugar, protein, etc. in the leaf, then definitely it is going to enhance the level of the osmolytes like proteins, etc., tree hallux sugar, etc. in the grain also. And I think that it's my opinion that the grains definitely they are going to be bold and these biochemical parameters are definitely going to be enhanced. And this is my preview that because you can see here that in this slide that in this also you can see here that the shoot, root biomass, etc. They are very much enhanced here, and also the chlorophyll content is enhanced. Then photosynthetic rate is enhanced. Then definitely food, etc., protein, etc. They are going to be more synthesized in the grain also.
Madhulika, what was the composition of your bacterial consortium that you used?
Yes, ma'am. I have used Azotobacter, Enterobacter, and Lactococcus, and these are the sources for the NPK. And I have used 20 gram of this bacterial consortium in 4 kilogram of soil, and this was proposed by the Microbiology Department IRI. From there, where I have got this.
You got the consortium from there?
Yes, ma'am. From IRI Microbiology Department.
Okay, fine.
Thank
okay,
you.
Madhulika, one more. Yes, sir. Yeah, go ahead.
Okay. Please, ma'am.
Okay, Madhulika, one question. What do you think? What is the reason that this bacterial consortium is improving to tolerate the stress?
Yes, ma'am. Thank you so much, ma'am. This is a very nice question. I have explained this already in my slide that these are the roles of the bacteria, that they help in the osmolyte production, nutrient uptake, phytohormone production, removal of ROS, ion regulation, and soil aggregation. And because of these roles of the bacteria, these bacteria are helping elevating the salinity stress in the wheat variety.
Okay, so some protein is also involved in this process, right?
Yes, ma'am.
So are you planning to find out that what is the factor, or what are the protein or the genes are responsible for this enhancement in the performance of the plant?
Yes, ma'am. Yes, ma'am. Actually, this is the beginning of the work. Later on, definitely I'm going to find out.
All right. Thank you, Madhulika.
Yeah, thank you so much, ma'am.
Okay, surprisingly, it was the same question I'm going to ask. So I was going to ask. So it's okay. I got your answer. Thank you.
Thank you so much, sir.
Thank you so much, Madhulika.
Yeah, Madhulika, I want to ask one question. Have you studied how this bacterial consortium, they are bacteria, how they are helping each other? Have you studied that?
You are asking about the mutual symbiotic, or what question?
Yes. How they are? Yeah.
No, ma'am. No. I have that point. I have not studied.
Okay.
I'm just studying I'm studying whether the fungi or the bacterial consortium is helping elevating the salinity stress in HD 2967 wheat variety.
Okay, fine.
Better I'm studying. Okay.
Okay, thank you so much, Madhulika. Our next participant is Anika Dadwal. Anika, are you there?
Yeah, hi. Am I audible?
Yeah, you are.
And is my screen visible to you?
Yes, it is.
Okay. So, good afternoon, everyone. I am Anika Didwal, a PhD student from NSIT. And first, I would like to thank ICIBLS team for giving me this opportunity to present my work in this prestigious conference. So today I'm going to present my work on cloning and heterologous expression and in silico characterization of beta-glucosidase of Mycilopsora thermophila for its applicability in bioethanol production from lignocellulosic biomass. So to begin with a brief introduction, so this beta-glucosidase, are the cellulases enzyme which cleave the beta-1,4 glycosidic linkages which is present in the cellulose to generate the fermentable sugars, which can be further utilized in the production of biofuels. So there are three classes of cellulases, which is endoglucanases, cellobiohydrolases, and beta-glucosidases. So three of these enzymes, they act synergistically in the conversion process. So I am taking this gene from a thermophilic fungus, which is Mycilopsora thermophila. It is a thermophilic fungus which grows optimally at 45 degrees Celsius, and it is a huge reservoir of cellulolytic enzymes. So in the genome of this fungus, there are total 9 BGLs. So only 2 of them as of now have been cloned and characterized. So I am working on the third one out of this 9. So one of the major limitation with this enzyme is it get inhibited with its product that it forms, which is glucose. So there is a need for like the production of this enzyme with high catalytic efficiency and its tolerance toward glucose. So thinking all these things in mind, we have devised these two objectives for my work. So first one involved the cloning and heterologous expression of this BGL gene of the fungus, and later on its characterization and structural analysis using various in silico approaches. So this involves the methodology involved for this research work. So this one is the cloning so I cloned this enzyme, this gene into PET28 vector, and then I transformed it E. coli and produced the recombinant anti-BGL. And then later on, I expressed and purified the protein and analyzed it for its enzymatic activity. So this for the objective 1, and for the objective 2, this was the methodology that I adopted. So the protein structure was modeled and in silico characterization was done, and also the docking and MD simulation were also performed. So coming to the results, so we used the synthetic MTBGL. We got it synthesized because I tried to also clone and express the wild type, but we were not getting any expression into the wild type one. So we got it synthesized and we transformed it into the BL21, and I screened for the positive clones using colony PCR and double digestion. So as you can see in this, this is the production of the recombinant. So the lane 4 is showing the successful expression, and 2 and 3, they are the controls. And so this one protein was further purified using his-tag column chromatography. So this was purified. I performed the enzymatic assays to check for its activity. So this is the qualitative assay which shows so this is actually a MUG assay, which is known as 4-methyl amberiferyl beta-D glucopyronoside, which is a substrate for this enzyme. So it is present in the plate, and as you can see in these 4 wells, so I poured in the enzyme were poured in these wells, and in the center it is the control. So when the plate was observed under UV, there was no fluorescence in the control, whereas you can see the fluorescence is quite evident in the wells with the samples. So, and also the optimum enzymatic activity was performed, which says that the pH this MTBGL was active. Optimum activity was observed at pH 8 and temperature 60 degrees Celsius. And the in silico characterization was performed using the ProTparam tool of the XPESI, which is the in silico strategy. So I computed the molecular weight, PI, atomic composition. II, AI are basically the instability and aliphatic index, which suggests the thermostability of the enzyme. Gravy suggests the hydrophilicity of the enzyme. So this is just the physicochemical parameters that I assessed. As I said that the protein structure and it has not been characterized, so I tried to model the structure of the protein using a modeler software which runs on a Python script. And the built model was further validated using various online softwares, which is Rampage, Verify 3D, iWrite, and so on. So all these scores suggest that the protein model is of acceptable quality. MD simulation run were performed for 100 nanoseconds, and you can see in this figure that the protein structure was quite stable at just after 20 nanoseconds. So there was no variation was observed in this. So the protein structure was quite stable, and the active site analysis was performed using the sitemap analysis of the Schrödinger software. So these are the top 5 potential ligand binding sites that were identified. So based on the highest site score and the volume, site 1 was selected for further docking studies. So I performed the docking with the substrate as well as its inhibitor to see the interaction between both of these ligands. So this is the 3D representation of their interaction. So let me just better explain you with this. This is the 2D image of the earlier 3-dimensional image. So this magenta arrows, they are representing the hydrogen bonding with these amino acid residues of the MTBGL with the cellobiose and with the glucose. So this table is showing the dock score values of the substrate of MTBGL, and this is the inhibitor. So as you can see, the dock score value of the substrate is more negative for the substrate, whereas it is quite less negative for the inhibitor, which suggests that the protein is having more affinity for the substrate as compared to its inhibitor. And in the interacting residues, if you see clearly that these are the highlighted ones, they are the common residues which are involved. So we can say that this is the product inhibition. Because of this, we can say that the glucose may not bind to these residues because the substrate is already binding to it. So this docking study suggests that this enzyme could be a glucose tolerant enzyme. And we have also identified the catalytic amino acid residues with the multiple sequence alignment. So this was performed, and the red arrows representing the conserved amino acid residues that are having a role in the ligand binding, whereas this triangle shaped and the star marked are the residues which are having a role in the catalytic activity of the enzyme. So
please wrap in a minute.
Yeah, sure. So the study concluded that the enzyme is active at it's thermostable. Its optimum activity is at pH 8 and 60 degrees Celsius, making it useful for the saccharification of the biomass and its utilization in the bioethanol. And the 3D structure of this enzyme is being proposed. Also, the amino acid residues, they have been identified, which played a catalytic important role in the substrate binding. And the docking analysis also suggests that this enzyme could be one of a glucose tolerant enzyme. So therefore, overall, the data suggests that this enzyme we can utilize it for various industrial processes. And these amino acid residues that we have identified, we can further play around with them to use it in further enzyme engineering process to attain a further high catalytic efficiency. Thank you so much for your kind attention.
Thank you, Anika. Any questions from judges?
Yes, I have a question, Anika.
Yeah, ma'am.
So, one of the slide you have shown a plate assay for enzyme activity estimation, right? So how you exactly quantify these activities?
Ma'am, this is the qualitative one. The quantification is usually done with the PNPG, which is paranitrophenol glucopyronicide.
So here you did just with the
Yeah,
qualitatively, not quantification.
Yeah, no.
So how you determine that what is the intensity of light, what is the basis for this graph? You have shown the activity graph.
Yeah, ma'am. This one is with the PNPG, which is we observe the UV absorbance of the samples under 405. So the substrate is not this one. For this activity, I have used PNPG, which is a paranitrophenol beta glucopyronicide. So which is a substrate. So I incubated this enzyme with the substrate at 50 degrees Celsius, and then I observed the UV absorbance. So the absorbance was high on this when we use this pH at this temperature. So that is the substrate that I used for this, but this is just the qualitative one.
Okay, yeah. Thank you for clarification.
Yeah, ma'am. Thank you so much for your question.
Okay, thank you so much, Anika.
Thank you.
Our next participant is Karthike Saxena.
Hello, am I audible?
Yes, you are.
Yeah. Let me share my screen. Is my screen visible?
Yes, it is.
Yeah. So let's start. Okay. So hello everyone. I'm Karthike Saxena, the lead and presenting author of the project entitled Immunohistochemical Analysis of Telomerase Reverse Transcriptase TERT in Conjunctival Melanoma using trichloroisocyanuric acid solution as a bleaching agent. So there are three emphasizing terms in my title: telomerase reverse transcriptase TERT, melanoma, and the trichloroisocyanuric acid. So the TERT is a protein coding gene, provides instruction for making a component of an enzyme called telomerase. It maintains telomeres, the repeated segments of DNA found at the ends of the chromosome. And it has been observed that telomerase is abnormally active in most cancer cells, which grow and divide without control or order. So basically my project is revolved around the association of TERT with melanoma. Melanoma is one of the most malignant tumor of the eye and conjunctiva. And the overexpression of TERT has often been reported in various carcinomas, leading to an increase in the activity of telomerase enzyme, which is crucial for survival of cancer cell. TERT promoter mutation are frequent in conjunctival melanoma. However, its expression has not been reported in it. So the problem associated with the project which I needed to tackle were the excessive amount of melanin pigment in melanomas, which interferes with DAP-based immunohistochemical assessment. Second, melanin pigment and DAP both are of same color, that is brown. And the standard use of KMNO4 and K2CR27 as a bleaching agent has often been shown to alter the specificity and the sensitivity of the antigenic epitopes of the tissue, thereby hindering ISC-based investigation. So in order to resolve these problems, the main objectives of our study is to evaluate the use of trichloroisocyanuric acid solution as a cost-effective and reproducible technique of melanin pigment bleaching from melanoma tissue for immunohistochemical evaluation of TERT in conjunctival melanoma. So we took 10 samples of conjunctival melanoma, processed it and analyzed it with the HMB45, Ki67 and TERT antibody. Here you can see the protocol of TCCA bleaching I standardized for the experiment. The importance of timing is really a crucial in this experiment because if it put the sections in the TCCA solution for so long, it will affect the counter staining. And if we do the process for short duration, the effective bleaching of melanin pigment won't be achieved. We need to stick to the timings for better results. The possible chemistry of bleaching the melanin pigment might be a work of hypochlorite ions. As you can see in the figure, these ions generating from the trichloroisocyanuric acid and further breaking down to forms a nascent oxygen, which is responsible for breaking down of melanin ring. But still, further investigation and experiments are required to validate the chemistry of bleaching with TCCA. So here's my finding. You can see in the figure 1A, it is a section of conjunctival melanoma in which a melanin pigment is dense and prominent. The following figure 1b is the control section of conjunctival melanoma before TCCA treatment, counter-stained with hemotixylin stained, showing the melanin pigment. And in the figure 1c, you can see the magic happened. And the whole melanin pigment is gone when I treated the section of conjunctival melanoma with TCCA solution. So as we know that TERT is a research antibody. So firstly, I tried the immunohistochemical assessment with steeple antibodies like Ki67 and HMB45 to check whether it is affecting the antibodies or not. In figure 2a, the section of conjunctival melanoma, you can clearly see the beautiful HMB45 cytoplasmic positivity. And in the figure 2b, the nuclear positivity of Ki67. Through this, I inferred that it is not affecting the immunostaining. So now I could use this for the research purpose. In figure 3a, it is a section of conjunctival melanoma TCCA treated in which you can see the TERT cytoplasmic positivity. And in the figure 3b, where you can see the TERT nuclear positivity in the section of conjunctival melanoma after TCCA treated. So yeah, my standardized protocol worked. So here are the results of the project that with the help of 5% TCCA solution, we can achieve effective bleaching. HMB45 and Ki67 expression was found out in the 100% of the cases. TERT positivity was found in cytoplasm as well as in the nucleus. The nuclear positivity of TERT was observed in the patient with primary acquired melanosis, and the TERT expression was found out in 70% of the melanoma cases. To be succinct, I would like to conclude that the overexpression of TERT was observed for the first time in conjunctival melanoma using immunohistochemistry. Well, it is better than classical method of melanin bleaching with KMNO4 and K2CR207 because the bleaching effect of TCCA solution didn't cause any interference with immunostaining using TERT antibody. This study advocates the use of TCCA solution for effective and rapid removal of melanin pigments from paraffin-embedded sections to expose the epitopes for immunohistochemical analysis. So for the acknowledgement, this research work was carried out in Dr. R.P. Center, All India Institute of Medical Sciences, under the mentorship of Professor Seema Sen, MD pathologist, Dr. R.P. Center, AIMS New Delhi, guided by Dr. Perumal Jairaj, Assistant Professor, Department of Zoology, Shri Venkateshwara College, and consented by Dr. P. Hemlata Reddy, Principal, Shri Venkateshwara College. Thank you. Thank you so much for giving me this opportunity to present my work. Thank you. Yeah.
Thank you, Karthike. Any questions from judges?
Karthike?
Yes, ma'am.
Is the methodology reported in other types of melanoma besides this conjunctival? You have standardized in
Actually, I'm still working on standardizing this protocol for choroidal melanoma. Firstly, I tried this on conjunctival melanoma, and now I'm trying to do this for choroidal melanoma. And I think it has been reported for ovarian melanoma. I am not sure. I have to look into it.
Okay. What are the other techniques that are reported?
Bleaching methods like KMNO4 and K2CR207 has been reported, and H2O2, ma'am. H2O2 peroxidase we can use, but it takes a lot of time to de-bleach. It takes around 24 hours. And for the KMNO4 and K2CR207, if we use, it bleached out completely dab. So, ma'am, that's why I use TCCM.
Okay.
It doesn't show any effect on tissue antigen, or DAP.
Okay, fine.
Thank you, ma'am.
Thank you so much, Karthike. Our next candidate is Ragul.
Good afternoon, ma'am. Am I audible?
Yes, you are.
1 minute, ma'am. I'm sharing my screen.
Okay.
Um, Hello, ma'am. So for my side, I'm unable to share my screen, ma'am. So can you check? Is there any problem?
Yeah, just a
The
second.
required questions have been given. Please try again.
Hello.
Yes.
Ma'am, it is not opening, ma'am. I cannot able to share my screen, ma'am. So host has to allow it. The showing for my side is like that.
Just a second.
So otherwise, is it possible to open from your side, ma'am? My PPT. I can go
Yeah,
ahead with that.
just share with us.
So already I sent the mail. Is if it is possible means you can share by your side, ma'am. So I'll go ahead with that one.
Just a second.
In the meantime, Nikita, you can call the next participant.
Okay. The next participant is Suchanda Agasti. Suchanda, are you there?
Yeah, are you able to hear me, ma'am?
Yes, I can.
Yeah, so I'm sharing my presentation. So, very good afternoon to all esteemed jury members. So, I am here to present on the topic regulation of inflammatory mediator uh, released by mast cells during allergic response by dynamic phosphorylation of T snare SNAP23. So, as we know that allergy is the most common disease which cannot be cured, and due to urbanization and change in lifestyle in the developing countries like India, so nearly 20 to 30% people are being prone to allergies. So it is very essential to look for better and affordable novel therapeutics which have minimum side effects and can target multiple allergic diseases. So mast cells upon allergen activation, they degranulate and they release various inflammatory mediators like histamine, serotonin, proteases, and also lysosomal hydrolysis like beta-hexosaminidase that initiate inflammatory response. And as we have described here that upon allergen activation, mast cells activate a signaling cascade followed Fc epsilon receptor cross-linking, and upon that, when the mast cells get activated, the granule and granule membrane fusion occur with the help of snare proteins, and in mast cells, different snare proteins involved in regulated exocytosis, including SNAP23, syntaxin 4, and VAMS, along with many other accessory proteins. So here we have already found that SNAP23 is a very a vital protein that is involved in mast cell exocytosis, and it is a peripheral membrane protein which is of 23 kDa, and it has a very important role in cell survival, developmental process, and also in normal cell functioning. And SNAP23 has 5 conserved cysteine residues and also to induce phosphorylation site, which has a very essential role in membrane association, also in controlling mast cell exocytosis. So SNAP23 has a very important role in granule membrane fusion during regulated exocytosis in mast cells, and it has been already been uh, previously it has been already been studied that the kinetics of SNAP23 phosphorylation mirrors the kinetics of exocytosis, and so we have studied that as we have already known that these two means induced phosphorylation residues, that is serine 95 and serine 122, has a very essential role in mast cell exocytosis. So we have studied the phosphorylation kinetics by Western blotting, and we have found that these two serine residues, that is serine 120 and serine 95, that the phosphorylation peak of serine 120 start from at 5 minutes and it continues till 10 minutes. After it gradually decreases, and at 45 minutes it is the dephosphostate. And but in serine 95 it start gradually from 10 minutes and it continues to 20 minutes the phosphorylation state, and gradually decreases, and 45 minutes is the dephosphostate. So it has been concluded that the serine 120 get phosphorylated first, then serine 95. And previously it has been studied that serine 120 may be affecting the signaling as well as the exocytosis in mast cells. And so we have also studied the exocytosis in transfected mast cells by HHLS assay, and we have found by transfecting this phosphomimetic and phosphonegative mutant and also the basal phosphorylation site mutant into the mast cells, and we have observed that there is a significant inhibition in net HGH release from transfected RBL mast cells in comparison to wild type SNAP23. So it indicates that SNAP23 phosphomutants affects mast cell exocytosis. So the main objective of this study is to study the role of phosphorylation at specific residues of SNAP23 in the intracellular localization in resting and activated mast cell. So what we have done is that we have transfected the all the negative and phosphomimetic as well as the wild type mutant into the RBL mast cells, and after 24 hours we have allergen activated it at 10 minutes and 45 minutes, because 10 minutes is the phospho related state and 45 minutes is the phosphonegative, so is the dephosphostate. And we have observed under the confocal microscopy, and we have seen that in the wild type at 10 minutes, that is the phosphostate, the SNAP23 it's internally located in the cytoplasm, and some it is found in the internal granules also. So as well as in other phosphomimetic mutant in the resting state, that is a serine 95D and 120D in the resting as well as in cross-link state, we have observed that the plasma membrane association decreases, and the mutants are internally located with the granules. So also we have observed in other two mutants, that is serine 95 and 120D as well. So we can conclude finally that SNAP23 plasma membrane association changes dynamically upon allergen trigger, and some seem to relocate to the intracellular sites. So to study further, so to exactly specify where the so to exactly specifically study the location of these phosphomimetic and phosphonegative mutants in the intracellular site. So what we have done is that we have stained the transfected mast cells with the LAM3 marker. So, and we have observed that in wild type transfected cells at 10 minutes and 45 minutes allergen cross-linking state, we have observed that wild type SNAP23 associates with the internal membrane LAM3 in cross-link stage, that is 10 minutes and 45 minutes, as we have already shown here. And also, but in phosphonegative state, we have observed that in the resting and as well as in cross-link stage, there is no association with the LAM3 marker. But in phosphomimetic state, that is double phosphomimetic state, that is 95D and 120D, we have observed that the 95D and 120D is totally internally associated with the internal membrane, that is the LAM3 marker in resting as well as in allergen cross-linking state. So also the same we have observed with 95 and 120D, but in this case we have not observed in the resting state that it is associated with the LAM3 marker, but in allergen cross-link state it is associated with the LAM3 marker, as well as in 95D and 120A also in allergen cross-link state. The mutant is associated with the LAM3 marker. So finally, we can conclude that the SNAP23 95 and 120D and vice versa SNAP23 95 and 120 most likely behave as SNAP23 wild type. So we can conclude that the interplay of serine 95 and 120 phosphorylation site may regulate the dynamics of SNAP23 membrane association as well as the regulated exocytosis, as we have observed. Finally, we can conclude that during allergen cross-linking, so SNAP23, which is associated with the plasma membrane, some are they move to the cytoplasmic region, and some also partly SNAP23 also relocates to the granule membrane, that we have shown from our study that it means relocates to the LAN3 associated membrane. So these two residues are very essential for SNAP23 membrane association and also in turn mast cell regulated exocytosis. So my future work is to just to validate by membrane cytosol experiment that whether it is internal totally with associated with the internal granule membrane or how much is remaining in the cytoplasm. Then further from by proteomic study in the transfected phosphonegative and phosphomimetic mutant, I can unravel the protein interacting partners. So these are the reference and publication. So I highly acknowledge my supervisor for consistent support and guidance, and we highly acknowledge all the funding agency for the financial support that is DST serve, that is on ongoing project UGC UPA to CSIR, and we are highly grateful and thankful to Dr. Paul Erosh from NIA so for providing us the antibodies, and we also highly acknowledge the ARF and CIF School of Life Sciences JNU. Thank you.
Thank you. Any questions from judges? Any questions?
So Chanda, what do you expect from the proteomics studies, protein interacting partners? What?
Ma'am, I can target those partners so that we can, after finding those partner, we can target it, and so that whether has any role in regulating mast cell exocytosis and all. So because the snare interacting partners, we have found from other studies that it has a role in mast cell exocytosis. And
can you give some examples of the interacting partners?
Ma'am, like RABs are there, so VAMs, synaptotagmines, and monks, other accessories and regulatory proteins. So it can help us.
Okay.
Okay, thank you, Sachanda. Our next candidate is Our next candidate is Ragul.
कैसा था राहुल? हाँ?
Ragul, are you ready?
Yes, ma'am. Yes, ma'am. This, ma'am, slide is visible.
No, it isn't.
It is not visible, right? I don't know why, ma'am. It is not visible.
Wait a second, we are presenting.
Okay, ma'am. Now it is.
Yeah, it is visible now.
Oh, okay. Thank you, ma'am. So, ma'am, can I start?
Yes, you can.
So good afternoon, everyone. So myself, Raghu. So I am here presenting my project entitled on preliminary investigation of Prosophis julivara wood-based regenerated biomaterial for defluorization of contaminated water in Tirupati region, Tamil Nadu, India. So basically, we know that nowadays scarcity of water is a very huge factor, and then there are many different factors and many different processes which helps to purification of water, like boiling, sedimentation, activated carbon filtration, and many researchers they are finding cost effective and regenerated biomaterial to remove many contaminants, because nowadays there are a number of companies they are releasing the dye like based products in the water. For that we should have to eliminate the thing. So in addition, the target is from my study is choosing this particular plant. Why means, so nowadays the government and many NGOs they are initiated to eradicate this particular tree like Prosophis julifera. It is nothing but in Tamil we would say that the Karuvela Maram and then Sima Karuvela Maram. This comes from abroad. There are many scientific reasons behind is that it takes more, it consumes more amount of water. For that, So the government itself, they were initiated to eradicate. So after the cutting of these particular trees, they simply throw it down, so make it a pollution. It's automatically degraded. But instead of wasting these things, how we can able to using this particular wood material to purify or deflorization of water. Normally, this particular plant has deflorization capability. So the modified method is that, so synthesis of crab shell chitosan from the crab shell by deacetylation process. So after the synthesis of these things, so this is the actual experimental procedure. So the thing what we have did, so initially we have cut into the wood pieces like a 2 mm, 3 mm, 5 mm, like that category. So afterwards we have incorporated, so cellulose based and then chitosan and silver nanoparticle, as well as the wood material. So after that, so what will happen means, these are the very different parameters. So RC means regenerated cellulose, and then RCCHAG means regenerated cellulose, chitosan, and a silver biocomposite. So which means, so initially I have taken a wood is a basic source. In that I have added the regenerated cellulose by dissolving the thiourea and the sodium hydroxide at the pH 7. So afterwards I have incorporated the silver nanoparticle in that solution. In addition, I have added the wood material, so which I have cutted. So after that, I have allowed into 30 degrees Celsius for over a time period to make it dry. So after that, I have a set an experiment procedure which I have mentioned here, like this setup. So the flow rate is which I observed is very effectively. I have observed this is the same image of RCCHAGW. Itself, it represents a wood biocomposite. It is a good effective material. So this is how the sample it look like. So before and after CCHAG. So determination of flooding. Initially, I have taken a 10 particular villages. So in that parcel, nearby villages is there. So that particular village contain huge amount of fluoride concentration. So by using this wood particular material, so elimination efficiency, which was determined by differentiating the wood thickness by mm. So the maximum efficiency was achieved by in 12 mm of wood material at 60%, which is nothing but normal level. So when we interrupt with vacuum pressure, so it will be very effectively it was achieved. So simultaneously, so what about the wood material which can able to absorb the water with allow into the contact time? So I have taken a particular that prescribed wood material. I have choken into the water. So from that I have evaluated that how much water the wood can able to absorb and suck, and then how can able to withstand in that particular water, because degrade wood itself, if we choke into the wood means it automatically will bulge or denature its properties. To avoid those things, to evaluate the property of this particular wood material is very important. So and also have agitation speed, because when we take in normal RO machine, there is some vacuum pressure is there, and then sponge is there, initial primary filtration and secondary filtration is there. So there are a number of processes there. So instead of that, so how we can able to make it in a simple method? So by using this method, there is a gradual increasing of the agitation speed when you increase the RPM and also the contact time. So this is the different pHs. So why I have tell means, so while we are adding the making the regenerated cellulose, almost I am dissolving it into the sodium hydroxide and thiourea, because it almost in a heavy base only. So when I am adding the HCl, I have to neutralize and it the 7 pH, where there I can able to observe the maximum elimination efficiency. The concluded that is that by using this wood material incorporated with the silver nanoparticle and chitosan and cellulose, which makes a good effective biomaterial for the deflorization of water. The future plan is that to eliminate the dyes, industrial dyes like chromium, CBB, like that and all, we have to planning to eliminate. So thank you.
Thank you so much for presenting. Any questions from judges?
So why only wood was used for biomaterial? Why only this was used as a biomaterial? Why not the other plant parts?
Ma'am, actually, so other plant material also it can be used, ma'am. But only one thing is that, so now government they are restricting to eradicate this particular plant, ma'am. In every lakes and villages, if you see, means they are strictly
Okay, that I understand, but
so and the main thing is that, so
the
naturally
government, you said that the government is eliminating the entire plant.
yes, ma'am.
No.
Hello.
Yes.
Hello.
Can you hear me? Yes.
Ma'am, voice is breaking, ma'am. I cannot able to hear.
Okay, Raghu.
Okay.
Hello.
Hello, sir.
Yeah, Raghu. I have a question that in your slide you have mentioned like you are using silver nanoparticle. Instead, there was zero valent ions are used. You are using both or separately?
So actually, first I have separated user because whenever actually initially my research only focused on the chromium elimination. So there I was achieved maximum efficiency. So after that, in my village in Tamil Nadu, Tirupati district, there was a fluoride toxicity, defluoridation, and dental fluorosis problem is there. So most of the people they are having the dental fluorosis and skeletal fluorosis. So for that only, so I am planning to decide. So for that only, this particular plant naturally it is having the fluoride absorption capacity. So ma'am asked, so why you choose like that question? So that is the thing, sir.
Okay, so you are planning to prepare a membrane or what kind of approaches you want to make
So actually,
for this kind of
I prepared
purification?
one biomaterial, sir, which is like a wood a coin like that. So 2 rupees coin is there, right? So like that, I have cut the wood material. So in that wood materials, I have incorporated these things there. So cellulose is a base, and chitosan is a act as a base. In that, so I have incorporated the silver nanoparticle and nano zero valent ion, so which act as a very effective material. So once I passed in that particular material water, so automatic normally if you give some amount of pressure means automatically the water can pass into the wood material. So but only one thing is that when I applied pressure, the elimination efficiency is increasing, and simultaneously the efficiency of water also increasing, because people they don't wait a long time to get more number less quantity of water. So for that only, if we allowed some amount of pressure means ultimately so we can able to drink, and then we can able to get sufficient water with less time.
So how your silver nanoparticle is helping in your purification of fluorides, I guess?
So
How?
actually, so when we take a wood material, so in that cellulose is there, sir. So the charge it is itself it is playing the major role. So fluoride, when we take a fluoride has a negative charge. So when we take a wood material, there is a different particle is there which is having the positive charge. For that only, so naturally that wood material is having or possessing the fluoride absorption capacity. So when I interact with these two material alone, instead of I am adding incorporating the cellulose, chitosan, silver nanoparticle. So I just have made an interaction between the raw wood material and that water. So there also I'm getting that much efficiency, but it's not when compared to I'm adding that wood materials.
Okay then. Thank you.
Thank you, sir.
One more question,
Okay,
Rukul.
Yes,
Yeah, you discussed that you are using cellulose from the tree material.
yeah.
So is it comparable to the pure cellulose? What you expect? Its efficiency?
Yeah, ma'am. So I have checked the purity of the cellulose also by so if you take a UV visible spec means there will be only one single peak which shows the purity, and also total
Hello.
and I have prepared both the things I have checked, ma'am. So when I purchased, so when I have prepared, so both the things are very same.
Okay. Another question. You have discussed TGA technique somewhere
Yes.
in the slide. So what you want to conclude from that? What can you say? What are you comparing?
How much stability it can, because normally when we pass into the water in that particular membrane,
No, that doesn't make the stability interpretation. Can you please open those slides? Hello.
This one.
Yeah.
Which one, ma'am?
The TGA curve. Yeah. So how you differentiate between these two?
So that is the thing, ma'am. So when we regenerated cellulose is there, and then regenerated cellulose, a chitosan along with the nanoparticle is there. So whenever we used to allow the water means there will be a temperature difference is there, because every time, so morning, afternoon, and mid time, three
Yeah,
categories.
that I got it. Yet I got it. That is your experimental procedure and how you have done
Yeah.
that. So what basically TGA explains? What is the basic principle behind? What you really looking for here?
Thermal, ma'am. Thermal property of the material.
Okay. So what you concluded from here?
So when compared to regenerated cellulose, the my recombinant, that regenerated cellulose, chitosan, silver nanoparticle will shows a good efficiency, ma'am.
Yes, but it is coming out to be near zero in your first part, right? So it
Yes.
should be or it should not be. What do you think
So
about it?
it should be, ma'am, because the stability should be maintained, right? So
Okay.
when we allow temperature, if it is increased, means the material should not be degraded.
Mm-hmm.
That should be some standard material should be there.
So
Some
here in this material, you have the cellulose, right?
Yes, ma'am.
Have you compared it with the pure cellulose? You are just taking the plant material.
No, ma'am, this is a preliminary. Still, the process is going on, ma'am.
Yeah,
So
that's
after this,
right. But
only
you
when
have
I'm
to,
prepared,
yeah, but you have to have a model experiment with that also, how it will be with your pure cellulose. So you can't
yeah,
really
ma'am. So
compare.
actually, I gave actually almost 17 samples I gave, ma'am. So I'm waiting for the results only. So that is the thing. Once if I get means I have discussed it, so it is not there. That is the thing, ma'am.
Okay. Okay. Thank you.
Thank you, ma'am.
Thank you so much, Ragul.
Thank you, ma'am.
So our next candidate is J. Priyanka.
Am I audible, ma'am?
Yes, you are.
Yeah, I'll share my screen.
Yeah, sure.
Is my screen visible?
Yes, it is.
Yeah. I'll start my presentation now. Good afternoon, everyone.
Yes.
Good afternoon, everyone. Today I'll be presenting one of our lab's research work where we have explored the therapeutic efficacy of a novel compound against triple negative breast cancer. Before moving on to my research, I'll give a small introduction about breast cancer. As we all know, breast cancer is a commonly diagnosed cancer among women, and it has led to 4.2 million deaths in 2017. Among various breast cancer types, triple negative breast cancer contribute to 20% of the overall breast cancer, and it is the most aggressive form of breast cancer, and it is very difficult to treat. Although chemotherapy is the standard treatment strategy that has been used for the treatment of TNBCs, over the past few years, there has also been a huge increase in the research or rather discovery of new treatment strategies for TNBC. But unfortunately, the major drawback of the existing chemotherapy is that it also causes a deleterious effect on the healthy cells. So we team of researchers have discovered a novel indinone derivative where we have used this to explore the efficacy of this NID against triple negative breast cancer. So here we have used two TNBC models, which is MDMB231 and MDMB468 in comparison to epithelial-like cells, which is WRL68. So to start with, we have treated the NID in MDMB TNBC cells and also simultaneously in WRL68 cells. We have seen that there was a dose-dependent decrease in the viability of TNBC cells, but interestingly, we couldn't see any decrease in the viability in WRL68. Further, we have, in continuum to that, we have also seen that there was a decrease in the expression of Ka67, which is a proliferation marker. So hereby, from these two experiments, we can say that this NID specifically reduced the cell viability of TNBC cells by inhibiting the Ka67 proliferation. Also, we have checked the expression of P65, which is a transcription factor and is responsible for maintaining cellular homeostasis, and also it is important for the survival of the cells. So here also we saw a drastic decline in its expression. Further, we know that P53 antagonizes P65. So there is a negative feedback loop which exists between P65 and P53. Here we saw that both the TNBC cells showed an increase in the expression of P53, but intriguingly, we saw that there was no change in the expression of the epithelial-like cell line, which is WRL68. Downstream of NF-kappa B, P65 is survivin. It acts as a transcription factor for survivin. Survivin, as we know that as the name indicates, it is essential for the survival of the cells. And also it is overexpressed in various cancer cells. More specifically in breast cancer, it is overexpressed. Here we see that since NF-kappa B, which is P65, is going down, survivin expression was also going down. Simultaneously, downstream of P53 is P21. We saw that since the expression of P53 is going up, P21 expression was also going up. So further, we explored what is happening to the cell cycle. We saw that most of the cells gets accumulated in the G1 phase of the cell cycle, which prevent them from entering into the S phase. We validated this result by checking other cell cycle proteins which is involved in the cell cycle. We saw that cyclin D1, which is involved in the completion of G1 phase of the cell cycle, was also going down. And if you see here, the cyclin A along with CDK2, which is essential for progression from G1 to S phase, was also going down. So from here, we concluded this results. We validate our cell cycle analysis result where we have concluded that it is inducing a cell cycle arrest. Further, we want to know what is happening to the fate of cells. So what we have done is we have done a SA beta gal staining where we saw that here, if you can see, there was a dose dependent increase in the SA beta gal positive cells in comparison to the positive control, which is doxorubicin. Also, we have treated it with pifitrin beta, which is an inhibitor of P53, to check whether the senescence is induced, which has been induced, is dependent on P53. So here you can see when we have inhibited P53, senescence was not happening. In both MDMB, this experiment was also replicated in MDMB 468. From here, we concluded that NID induced senescence in the triple negative breast cancer cell, only in the triple negative breast cancer cells, and which is P53 dependent. Interestingly, when we have treated in WRL 68 cells, it was not inducing senescence. Here you can see that senescence has not been induced on treatment with NID in comparison to the positive control. So in conclusion, what I'd like to say is NF-kappa B, which is a transcription factor, as we know that it antagonizes the expression of P53, which is a tumor suppressor gene. There exists a negative feedback loop between these two proteins. Downstream of NF-kappa B is survivin. This particular protein is overexpressed in various cancer, more specifically in breast cancer. So we also know that survivin also inhibits P53. Also, we know that downstream of P53 is P21, which is essential for the cell cycle progression. NF-kappa B, apart from involved in survivin expression, it also regulates the expression of cyclin D1, where we know that which is essential for the progression from G1 to S phase. Simultaneously, P21, what it will do is it will inhibit cyclin D1. On the other case, it also inhibits CDK2 and cyclin A, which is involved in the progression of G1 to S phase. So this is essential for cyclin D1, cyclin A is essential for progression from G1 to S phase. So when we have treated novel indolent derivative for 24 hours, we saw that cells are getting accumulated in the G1 phase of the cell cycle, which lead to senescence. I would like to conclude by saying that this novel indolent derivative is specific only to cancer cells by inducing cell cycle arrest and putting the cells into senescence. So I would like to conclude by saying that this NID essentially put the cancer cells to sleep. That is, in technical terms, senescence. Also, I'd like to say it is specific only to the cancer cells. Thank you. These are my references.
Thank you, Priyanka. Any questions from the judges?
Yes, Priyanka, which assay you have used to check the senescence in TNBC cells?
SA beta gal staining, ma'am.
Beta gal staining. Okay. Thank you.
Thank you, ma'am.
Priyanka, in cancer situation, P53 itself gets mutated. So
yes,
how do you distinguish that the P53 is regulating the cell cycle? And
actually, thank you for that question, ma'am. We also had that question. Actually, if you can see from our result, in this particular novel indolent derivative is specific only towards the TNBC. In both the TNBCs, P53 are mutant. Whereas in the WRL 68 cells, it is not mutant. So what we think is This NAD induces senescence in the TNBC cells which has mutant P53. Probably in other words, I can say in P53 which is not functional.
So how can you check it?
Sorry, ma'am?
How can you check it whether it is a mutant? It is just your probability. It is that you are saying that it could be so.
Yes, ma'am. Probably that is what we are thinking.
Yes. So, is there any way to check it?
Yes, ma'am. Probably we can screen for like what are all the mutants that is available in both the TNBC cells, and we can see whether there is an overlap in particular mutant, so that we can check in other cell lines also which harbor that particular mutant.
So, have you any plans to do so?
Yes, ma'am. This is an ongoing work, so we have done some preliminary studies. So this will be we will be proceeding with that, ma'am.
Okay.
Okay. Thank you, ma'am.
Next, we have Shreyas. You can start now.
Can you hear me?
Yes, you're audible.
Okay. Good afternoon, everyone. My name is Shreyas Kudwali. I'm a research scholar at Central Interdisciplinary Research Facility at Sri Balaji Vidyapeeth, Puducherry, under the guidance of Dr. Anita T.S., scientist CIDRF. Today, I'm going to present a part of my study that is titled Metformin and Epicatechin Gallate enhances the anti-glioma efficacy of temozolomide by attenuating PI3K-AKT-mTOR pathway. Coming to the introduction, glioblastoma is one of the most malignant of all brain tumors, accounting for more than 50% of all glioma cases. The incidence is just 2 to 3 per million, out of which three patients, 2 are men. Current diagnosis is PET, CT, MRI, and the location of the tumor could be frontal lobe, cerebellum, brainstem, and spinal cord. Current treatment strategies include surgical resurrection, radiotherapy, chemotherapy with temozolomide. Yet the average survival is just 12 to 15 months, which is the major factor for us to choose this particular malignancy. So one of the well-established in vivo models is the orthotropic xenograft induced glioma model. The strategy which we applied for this study was repurposing of drugs, which is actually a current strategy used not only in treatment of malignancies, but also in other diseases. The main reasons being it is faster than the conventional drug development studies and at the lower cost. So in this study, we employed metformin, an antidiabetic drug, as well as epigallocatechin gallate, a green tea flavonoid, in treatment of glioblastoma in combination with the standard drug. Hence, our objective was to determine the inhibitory effect of metformin and EGCG in combination with temozolomide on PI3K signaling pathway in a glioma-induced rat xenograft model. So coming to the methodology, 1 million cells were injected intracranially into the brain of the animal and using a stereotactic apparatus. After 20 days of implantation of glioma, the animals were randomly segregated into various treatment groups as shown in the figure. And the animals were subjected to various treatment via intraperitoneal injection, and the tumors tissue was isolated for various studies. The studies which we performed were survival studies, hematoxylin and staining, and immunohistochemistry was performed from angiogenic and proliferative markers. We also studied the gene and protein expression levels of PI3K markers and apoptotic markers. Further, to understand the mechanism of action, we also analyze the protein levels of VEGFR, a key regulator of this pathway, as well as caspase-3, another terminal protein of apoptosis. And further, we perform molecular docking. Coming to the results, we performed survival study using Kaplan-Meier curve, and it is evident from the graph that the triple drug combination significantly enhanced the survival of the animals beyond 25 weeks, whilst the tumor control animals managed to survive for a maximum of just 8 weeks. And we performed H&E staining, and from which is evidence that the triple drug combination also reduced tissue defragmentations and wall structures from formation, indicating a reduced tumor growth. And our immunohistochemistry staining show that the triple drug reduced the Ki67 concentration, suggesting a reduced proliferation. Further, even reduced VEGF shows that there was lesser vascularization in the tumor tissue. Coming to the gene expression, we studied the expression of biomarkers of PI3K pathway, basically PI3K, PDK1, and AKT, and also an antagonist of this pathway, PTEN. From this data, it is evident that the triple drug combination of TME significantly enhanced the antagonist PTEN and reduced the expression of PI3K biomarkers. And a similar trend was also seen in protein densities, which was performed from the Western blot bands. And coming to the expression of apoptotic markers, we saw that the triple drug combination significantly enhanced the genetic markers of apoptosis, and a similar trend was also seen in the protein studies. And surprisingly, in the dual combination of temozolomide and EGCG, also we found a significant increase in apoptotic proteins. So these were the bands which we got in our Western blot analysis. So to further understand the mechanism of action of our drugs in this particular pathway, we studied the expression of VEGFR and caspase-3, which are representative biomarkers for the pathways and apoptosis. So VEGF is the key regulator of this pathway, which binds to VEGFR, which then activates PI3K, leading to the entire activation of the pathway. Hence, we performed the docking study to study the interaction between our drugs and their metabolites along with that of VEGFR protein. This ELISA study gave us a surprising result that the drug combination significantly reduced the expression of VEGFR and enhanced the expression of caspase-3. So we employed the 3D structure of VEGFR here, and the drugs and their metabolites are as shown here: the TMZ and its metabolite MTIC, AIC, metformin and its metabolite guanylurea and EGCG. And to our delight, all our drugs and their metabolites showed good interaction with at the active sites of VEGFR, suggesting that inhibition of VEGFR activation of PI3K. So coming to the conclusion, our study has shown that the triple drug combination of TMZ, MET, and EGCG as an effective model for glioma treatment, glioblastoma patient. However, it still requires further clinical validation. And to acknowledge, I would like to thank Dr. Adityan C for his immense support in our study and for the Dr. Rani Adityan Fellowship. I would also like to thank Sri Balaji Vidyapeeth for providing us the instrumentation and lab facility. And also, we would like to thank Dr. K. Satyamurthy, Director of Manipal School of Life Sciences, for providing us with the animals and the animal facility. So these are some of my references for this study, and thank you.
Thank you, Shreyas. Any questions from the judges?
Okay, Shreyas, I have a question.
Yes, sir.
You have used metformin. So did you use any salt component with this metformin, or you use directly metformin?
It is pure metformin, sir. We studied.
Because sometimes it is noted that the metformin lowers the blood pressure in case of animal models or any system when we are using metformin directly. So have you come across such kind of?
So can you come again, sir?
Sorry?
I could not get you.
Okay. I said like metformin sometimes causes downfall of blood pressure generally. So for that, every time when we use metformins, they use some kind of salt together with that when they are prescribing or when they are being used in animal model or something. So did you come across something? when we were teaching with such?
No, sir, we did not do that study, and we did not analyze that.
Okay, so it is for the radiotherapy or chemotherapy, and which part?
It's chemotherapy, sir. It's
It's
chemotherapy.
chemotherapy.
It's
Because
actually an
generally
adjuvant that
metformin.
we did for temozolomide, which is the current standard therapy for glioblastoma.
Right.
So this we just wanted to study these two components as an adjuvant treatment, like along with the temozolomide in the chemotherapy.
Okay, thank you.
Okay, so thank you. Next, we have Sakti Jayasundar. You may now start. Are you there, Sakti?
I didn't know that. Metformin radio.
In case he's not there,
Maybe he is not present right now. Next, we have Saudipta. Is she here?
I can see Sakti now.
Just we have Sakti here. Next, it's you, Sakti Jayasundar. You may start now.
Hello, am I audible, ma'am?
Yes, you're audible.
Hello.
Yes, you're audible now.
Yeah, yeah. Yes. Visible, ma'am.
Yes, it's visible.
Can I start the presentation now?
Yes, you may start now.
Ma'am? Hello.
Hello, am I audible to you?
Yeah.
You may start now.
Student from Melmaruth Rajasthan Institute of Medical Science, focus enabling superior stability of intelligence. So, yeah. Shakti Jason Rajashekhar, first MBB workers enabling superior stability of COVID-19 mRNA vaccines using artificial intelligence. So the background of my work is the COVID-19 pandemic has brought the world to a standstill. So the only way we could move forward is with introduction of a vaccine. So there are several companies and universities throughout the globe working out for a COVID-19 vaccine with various other techniques. So one of them is the mRNA vaccine. So an mRNA vaccine at this point of time could be a relatively new frontier, which could have taken many more years to come into existence. But due to this pandemic, it's actually fast tracked, and we are working out for this vaccine. So this vaccine has got several pros. So whereas the main con is that its stability is really poor. So due to which it might not be possible to transport these vaccines and get it to the public so that they get vaccinated. So as of now, as you can see, the pandemic has infected many and taken the lives of many. So the aim of my study is to build a robust data model which could accurately predict the degradation rates of various RNA molecules when placed at various positions within the mRNA chain. So the methodology of my work, the data set of around 629 RNA molecules and their reactivity and various parameters like reactivity degree at a pH 10 in the presence of magnesium, without magnesium, and at 50 degrees with magnesium as well as without magnesium. So this data set of 629 RNA molecules is aggregated from the Kaggle repository. So I use this model, I use this data set to train various data models like GRU, LSTM, RNN. So the training to testing ratio which I have used is around 80:20. So this is the architecture of my work. So I take the mRNA vaccine data sets and I feed it through the gated recurrent unit. That's the GRU, recurrent neural network. That's the RNN, and the long short-term memory. That's the LSTM. So these three are basically A models. So I feed the data set to these models in pH 10 in the presence of magnesium without, and in 50 degrees with and without magnesium. So the results of my study, I have calculated the root mean square error, that's the RMSE values of predicting of my model, of basically the three individual models for predicting the three parameters with all the three models. So as you can see, these are the results for the three parameters using the three different models. So concluding, for the three parameters, the LSTM model predicts the reactivity with a better accuracy, while the RNN model predicts the other two parameters with a better accuracy. So individually, these models have been like the LSTM and the RNN model have been able to do well in the respective parameters. However, as of now, that's the work. But what we have to try to do is we have tried to hybridize these models. Let's hybridize the LSTM with the RNN network. So by hybridization, we aim to get a even better results for all the three parameters. So this model, I believe, could help the computational biologists build a mRNA vaccine with a superior stability. So that's it. Thank you.
Thank you, Sakti. We may now have questions if there are any.
Yes, I have a question. Can you hear me?
Yeah. Yes, ma'am.
Can you hear me?
Yes, ma'am.
So my question is, Sakti, Pfizer has used mRNA vaccine. Have you tried to compare your model prediction with the mRNA they have used for the COVID-19 for coronavirus?
Actually, this work is done like it's a challenge from the Stanford University, ma'am. Actually, so what they have thrown out a challenge in the Kaggle community. So what they did is like basically they provided a public data set of 629 mRNA data set values, and they have some other like 300 mRNA. So they test this for public. Like we can access these 629 RNA data set. So they take our code, they try it out with the other 300. So if they feel our model is better, they would try it out, experimentally. Like they'll create a physical model of the mRNA chain which we predict, and they'll physically test it out for its, the parameters like reactivity and other parameters.
Okay, I
So
got
as of
that was a Kaggle challenge then. All right,
yes,
thank you.
yes.
Thank you.
Yes, Sakti, what can you state regarding the stability of vaccine according to your model?
The RMSE value has been quite good, ma'am, comparing our model. That's I have only tried out with the preliminary models. That's the basic. These are the predefined models basically. So the LSTM and the RNN. So I like the next step of my work is like I aim at hybridizing these two. Like basically these are predefined models. So I just code it. So next I look upon a algorithmic modification of these two, so that I could bring out a hybridized model which would naturally be with superior characteristics. That it would have a still reduced error in predicting these parameters.
Okay, thank you.
Thank you, Sakti. Next, we have Niharika Gupta. You may present now.
Am I audible?
Yes, ma'am, you're audible.
Okay, fine. Is my screen visible to you all?
Yes, ma'am, it's visible.
Okay, just a sec. Yeah. So good afternoon, everyone. Um, Today I'm here to present my work on carbon nanomaterial based conductive ink for flexible paper-based biosensors. So I'll be as precise as possible with the presentation. So as a background, papertronics is an emerging field under the subhead of flexible electronics, and it deals with the development of flexible and foldable paper-based electronic devices such as biobatteries, fuel cells, and biosensors also. So particularly in regard to clinical diagnostics, paper-based biosensors have emerged as very promising tools in terms of their speed of analysis and sensitivity also. So just in case, like some of you are not very well versed with biosensors, this image on the right is, just a second. This image on the right is basically a basic schematic of a biosensor. So it basically integrates a transducing surface that can be optical wire or a piezoelectric crystal or even an electrode to a variety of biomolecules ranging from antibodies to even whole cells to analyze or to detect quantitatively various different types of analytes and molecules. This transducer is further connected to signal processing units. So in case of electrochemical devices, the transducer that we have is generally an electrode that converts our biological signal into electrical or electrochemical signals. And in case of paper-based electrochemical sensors, this transducer assembly is nothing but a paper substrate which is being modified by certain nanomaterials. So here on the left that you see is basically a general setup of an electrochemical biosensor. It is nothing but an electrochemical cell, but with 3 electrodes. This working electrode here, mostly in paper-based biosensors, we have usually a paper substrate with biomolecules and other transducing material on it. And then we have the reference electrode, which is usually AgAgCl electrode, and a cathode or a counter electrode, which is a platinum wire usually. These all are dipped in a buffer, which can be a phosphate buffer saline in case of biosensors or any other, depending upon the experiment. This whole assembly is then linked to a potentiostat that really records the signal and amplifies it for display. Now, in the case of paper-based biosensors, the ease of use is that we can translate this whole big assembly to a single piece of paper. Say in this thing, it's like 3x1 size of paper on which the whole 3 electrode assembly is here, the counter, working, and reference. So this is the main advantage of using paper sensors. So there is the other advantage is that these are cost effective and disposable, and then they can also be eventually utilized in point of care analysis also. So the main challenges because of which paper-based biosensors usually lag behind other mainstream biosensors is that with paper, we have to, particularly for electrochemical applications, we have to get sufficient conductivity on paper, and then we have to prevent the swelling of paper fibers also, because it has to be dipped in the buffer constantly for measuring. And another problem with biosensors particularly is the biomolecules, the material transducing surface should also be such that we can easily and robustly immobilize our biomolecules on the surface. And then obviously then there's the issue of reproducibility also. So in this study, what we have aimed is to develop a conductive ink that is based on carbon nanomaterials for printing on paper substrates, and eventually develop an electrochemical biosensor based on that. So in this particular study, I have used carboxylated multi-walled carbon nanotubes. So these are cost-effective carbon nanomaterial that are scalable, processable, and carboxylation not only enhances their solubility in various solvents, but they can also be used for efficient immobilization of biomolecules. And screen printing, the reason we are developing a conductive ink is because it would improve the batch reproducibility of the biosensing platform as such. The target or the analyte that we are going to detect using this biosensor is pathogenic DNA. To be more specific, it is the OPA gene sequence of Neisseria gonorrhoeae, which is the causative agent of gonorrhea. The main strategy that is being used in this study for detection is sandwich hybridization assay. So we have basically designed one capture and one detector probe against the a specific gene sequence of Neisseria gonorrhoeae OPA, and then used it for electrochemical analysis. So in brief, the methodology is simple. For preparing the ink, I've used an emulsion type of ink wherein an organic phase is mixed with deionized water, and a binding agent at a particular ratio is mixed, and eventually dispersion of carboxylated MWCNT is made. Then this emulsion is coated onto paper strips that are then further cut. And for biosensing studies, avidin biomolecule was immobilized onto these paper electrodes using EDC and histochemistry. So for the studies, I've done electrical conductivity studies, viscosity studies for screen printing purposes, cyclic voltammetric studies for electrochemical characterization and biosensing studies. So in this cyclovoltammetry, I have again used the standard platinum as counter, AgAgCl as reference, and I have done these studies in PBS buffer supplemented with potassium ferroferricyanide as redox mediators. So for the final biosensing studies, what I've done is first I've taken equimolar concentration of capture probe, which is biotinylated at 5' end, the target sequence, and the detector probe, which is tagged with methylene blue dye at the 3' end, and I mixed them in aqueous in equimolar concentration at this temperature, 56 to 58 degrees Celsius for 5 minutes. So after this incubation, I drop casted 25 microliter of this DNA mixture onto my avidin modified paper electrode, and then again incubated it at room temperature, say 25 degrees Celsius in humid conditions. After these, I wash them. Yes. Okay, fine. Sorry, please, what did you say?
Please wrap up in a minute. It's already been.
Oh, okay, fine. I'll just be fast then. Okay, this is the schematic I'm just going to. So the results are here. So for the conductivity studies, the maximum conductivity that I found was 2.47 Siemens centimeter, which is more than 10 raised to the power 4 times greater than the conductivity of bare paper. Then this is a small video. I'll just play it. I don't know if it is playable, but it can't be. So okay, I'll just leave it because it's sort of long. Let it be. So for the rheological analysis, within the shear rate of 0.1 to 1000 per second, the viscosity change was linear in proportional to the shear rate, which shows that it is thixotropic in nature and which is actually suitable for screen printing. Electrochemical analysis. This is the basic CV curve for the paper electrode and the Avedin modified paper electrode. So there is a decrease in the current, the peak current. Why this is? Because Avedin is a biomolecule that basically leads to decrease in the flow of electrons to the electrode. So this sort of confirms that Avedin has been immobilized on the surface. These are the biosensing studies that that I conducted. So here in this first graph, we see that with increase in the concentration, the peak current is really increasing because I've tagged my detector probe with methylene blue. That is also a redox mediator. And this is like at lower concentration, the methylene blue activity is not really detectable, but at higher concentrations, it is actually detectable. I can even get the redox peaks for that. This is the linear calibration curve that I got from it. So the linear range of the biosensor is 1 picomolar to 1 nanomolar. The sensitivity was 11.034. I'm sorry, I didn't write the microampere per log concentration. And the lower detection limit was found to be 10 picomolars. So in conclusion, I can say that we have developed a carbon nanotube based conductive ink that was coated onto paper and then eventually developed into a biosensor for a detection of pathogenic DNA. It was found to have good enough conductivity and the linear range of the biosensor was good enough. And particularly now we are going through optimization experiments to further enhance the conductivity while retaining the binding of the ink and also to work on the improving the sensitivity of the biosensor. Thank you. These are my acknowledgements. I acknowledge DTU for financial assistance, my supervisors, D. Kumar sir, DD sir, and Asmita ma'am, Jay Gopal sir, head of our department, all the faculty members, all my lab members, and all the PhD students that have helped in some way in this work. Thank you all. These are some of the references. Thank you.
Thank you, ma'am. Any questions from the judges?
Okay, Niharika, I have a question. You used multiple wall carbon nanotubes, right?
Yes, sir. Yes, sir.
So actually, it has both metallic as well as semiconducting properties. So how did you separate it?
How did I separate the metallic and?
Separated it. Semiconducting.
No. The multi-wall carbon nanotube that I have are commercially acquired. The carboxylated.
Okay. You take it directly
yes.
from industries.
Yes. They are commercially acquired. I did not.
Okay. So did you, did any experiment to confirm the uniformity of biotin on the surface, or did it help to, or did it alter the result, that uniformity of that biotin on the surface of your transducer?
Uniformity of Avedin, like I have immobilized Avedin on
biotin,
the.
right?
Sorry, I'm using.
You're using biotin,
yes.
Right? Okay, so you showed me the very good results related to biotin. I just want to know what was the uniformity? Did you show any kind of 10 graph or something?
Oh, the uniformity studies. Actually, for the morphological studies, I have to sort of conduct SEM experiments also and also AFM. So for that, I'm in conversation with other labs, other institutes in IIT Delhi and Delhi University. And they have a big backlog there. That's why those results are not there yet, but I'm in waiting. So I am for the uniformity studies. I'm on it actually, and basically we are in the process of scaling up the ink also. So we'll be screen printing it, and then also we'll be going for uniformity studies.
Okay, Niharika, you are using any kind of plasticizer to get it uniform pasted on the paper?
Yes. There is a binder in there. Yes, there is. Not particularly a plasticizer. I'm not really allowed to reveal that right now unless until it is published.
That's your. I understand.
Yeah.
Means like
Yes,
did it
yes.
hinder the results related to that, the quantity of your plasticizer?
Yes,
It is
yes,
affected your
yes, I did.
results?
Yes, exactly, exactly. That is the point that the process, the optimization basically, because we have to have a certain amount of binder in our ink. Otherwise, the ink would not adhere to the paper substrate. And at the same time, if we add too much binder, it would be so insulating that we'll get no conductivity on the paper. So that binder to carbon nanotube ratio, we are still in the process of sorting this out. How do we increase the conductivity while retaining the binder ratio? Maybe so. Yes. So I was already conducting these experiments. Like there are a lot of failed experiments behind this study. This is one thing that I successfully got done, and now again, this it's still in process. The optimization of the ink.
Okay, Niharika, there was very good results. Thank you.
Thank you, sir.
Thank you, ma'am. This is the last call for uh, K. Arul Kumar and Swadipta Pramanik. If you are present here, you may raise your hand. We now have Parasamal S. You may
Hello,
present
am
now.
I audible?
Yes, you're audible.
Okay, I'll share my slide now.
Yeah,
Okay.
sure.
Is this visible now?
Yes, it's visible.
Okay.
You may.
Good evening, everyone. I am Parasmal. I am here to present my research work, The Challenges in Diagnosing Infections by MPT64 negative Mycobacterium bovis BCG strain. So, as we all know, tuberculosis is one of the top 10 major cause of death worldwide. WHO report says in the year 2019, around 10 million people report to have TB globally, and among them 1.2 million are children. So, to prevent tuberculosis, live attenuated BCG strain was introduced in the year 1921. So, the WHO has recommended BCG vaccination for all the children at birth to prevent extrapulmonary tuberculosis in high TB burden countries. Even though the vaccine is safe, the administration may lead to some complications from mild to severe, and the complications are include BCG, lymphadenitis, fasciitis, and etc. So the incidence of complications of BCG infections are ranges from 2 to 1,000 per million vaccinations. The complications are associated with the residual virulence of the BCG substance. That is like some strains are more reactogenic than the others, and the viability of the vaccine, proportion of the live and dead bacilli present in the vaccine, and the dosage of the vaccine. So the mild complications, it may not require any treatment, but in case of severe complications, it requires a chemotherapy with anti-TB drugs. And since all Mycobacterium bovis strains are intrinsically resistant to pyrazinamide, CDC recommends the triple drug regimen consists of rifampicin, isoniazid, acid, and ethambutol. And so, as I said, the complications are mainly because of the residual virulence of BCG substance. Here we see the refined genealogy of, sorry, just a minute, refined genealogy of BCG vaccines. So, if you see, the BCG is derived from a virulent Mycobacterium bovis strain. Okay, so this BCG lost its virulence because of number of passages, and the attenuation came because of the BCG was the loss of the protein secretion system EX1, and this is absent in all the BCG substrains and due to the deletion of RD1 region. So this BCG strain is widely distributed among the countries, and this leads to the diversification of different BCG substrains, and thus dosage strains have been widely studied using the comparative genome analysis, and discovered that the numerous deletion and insertions of the genomic sequences, and also the signal nucleotide polymorphisms. So mainly, the BCG strains differ in the presence or absence of MPB64 gene and the number of copies of IS6110 gene. This MPB64 gene is widely code para 24 kilodalton antigenic protein, which is widely used as a diagnostic marker for differentiation of Mycobacterium tuberculosis complex infection and non-tuberculosis Mycobacterium infection. So most of 90% of the BCG vaccines are BCG Pasteur, Danish, Glaxo, Tokyo, and Russian strain. Among these, Pasteur and Danish strains are considered as a strong strains, and the Glaxo and Tokyo are considered as weak strains. So, however, in India, the public sector uses Danish strain, which lacks MPB64 gene, and some private sector uses Russian strain, which has MPB64 gene. So this leads to the challenge in accurate identification when it comes to the infection, reinfection. So how do we diagnose TB in normal? So currently, these are the available diagnostic procedure for TB detection in the clinical laboratories. This is considered to be a gold standard method. So initially, the specimen processing followed by the microscopic observation and BACTEC MGAT mediated culturing. So if once the growth is present for Mycobacterium, SD BioLine MPT64 card assay will be performed to differentiate the non-tuberculosis Mycobacterium and the Mycobacterium tuberculosis complex. So here the problem comes. If as I said earlier, some of the BCG strains has this MPB64 gene, and some of the strains doesn't have this MPB64 gene. So this leads to the complication of diagnosis. This MPB60 card negative BCG strains are misdiagnosed as NTM infections. So here we present 2 cases of BCG infections that are due to Mycobacterium bovis BCG strains, which are negative MPT64 antigen. So in the case 1, a female infant of 4 months old, and in case 2, a 1 year and 5 months old of a male child, they had extrapulmonary infections like axillary region swelling and the swelling of soft tissue on the knee joint. So these are the diagnostic methods that we initially perform to identify the cause of infection. The microbiological identification gave positive for Mycobacterium. That BACTEC MGAT gave the positive results for Mycobacterium. Followed by we performed the PCR and ICT rapid diagnostic things. Gene expert gave the positive for Mycobacterium tuberculosis, and the clinical isolates were rifampicin sensitive. But the MPT64 card assay given the negative result for MTVC. So hence we decided to move on with the molecular diagnostic methods, and based on the molecular way, we decided to speciate the clinical isolates. So these are the conventional PCR methods that we used to speciate the clinical isolates. The figure image A shows a schematic representation of RFLP pattern. So a single nucleotide polymorphism is exists among MTBC, particularly in GYRB gene. So if we perform a PCR of this gene and then followed by restriction digestion with the TAC1 enzyme, so we will be getting the band patterns like this for Mycobacterium tuberculosis complex organism. So this is the results that we got, the figure image B. So the lane 1 is we have used as a control, H37RA tuberculosis strain, and lane 2 and 3 are commercially available BCG, Russian and Danish strains, and lane 4 and 5 are the clinical isolates. So if you see, the clinical isolates gave the RFLP pattern as similar like the
Please wrap up in a minute.
Okay, similar like the other commercially available strains, but we are the pattern. So we decided to move on with the further PCR analysis with RD1 and RD2 PCR. So here we confirm you can see the band patterns. The 3 and 4 are the commercially available strains, and 5 and 6 are the clinical isolates. So we found this is a BCG strain, and the clinical isolates were confirmed as a Mycobacterium bovis BCG. So we to know which strain that caused the infection and which strain this is to MPT negative 64 is negative, we did the sequence analysis. You can see the single nucleotide changes is present in the Mycobacterium bovis and Mycobacterium bovis BCG among the Mycobacterium tuberculosis complex. So this is the electropherogram of the clinical isolate. After the sequencing, we did the blast analysis and we found that 100% sequence similarity with the Mycobacterium bovis Danish 1331 strain. Hence, the case 1 was identified as BCG adenotitis and case 2 was identified as BCG osteitis. So in conclusion, so as I said, in India, there are two different strains have been used. The use of two different strains for vaccination in the same population, one with and another without MPB64 gene, further complicates the problem of identification. Our finding emphasizes the importance of careful use of MPT64 kit with clinical correlation. That is gene expert results and also the mycobacterial growth morphological conditions, everything. And clinicians should also be aware that the finding disseminated BCG in young children is a hallmark of serious immune deficiency, which would need investigation and patient management. Acknowledgement, we are grateful to Amrita Center for Nanosciences and Amrita Institute of Medical Sciences for providing an infrastructural support, the financial support. Thank you. Thank you.
Thank you, Parasamil. Any questions from the judges?
Arisamal, what is the reason behind using Guy gene for the sequencing purpose?
That has been already reported, ma'am, by Casey H et al. So they have used this GAIRB gene to differentiate the.
There must be some reason for this.
Yes, ma'am. They use this one to differentiate the Mycobacterium tuberculosis complex strains. This complex includes tuberculosis, bovis, africanum, and caneti. All these strains causes tuberculosis. In order to differentiate those strains, they have used this GYRB gene and with the RFLP pattern.
What does this
So
code for?
we chosen this one.
In code for which enzyme?
Gyrase enzyme.
So isn't there any other way to see, differentiate these strains?
Yes, ma'am. This the second one, this RD1 and RD4. Here we could differentiate in the RD1. We could differentiate the Mycobacterium tuberculosis and BCG, and in the RD4 we can differentiate Mycobacterium bovis and Mycobacterium tuberculosis.
Okay.
Thank you, Parasamil. Next, we have Sadipta Pramanik.
Hello.
You may present now.
Am I audible?
Yes, you're audible.
Okay. Good afternoon, everyone. I am presenting my work entitled Reduced Cystathine Beta Synthesis Activity in Cigarette Smoke-Treated Hepatocytes is a potential cause of foam cell formation. So, what is foam cells? Foam cells are lipid-loaded macrophage cells that can form plaque. Plaque build up narrows arteries and makes them stiffer. That can lead to atherosclerosis and that trigger heart attacks and strokes. So cigarette smoke causes various diseases like COPD, atherosclerosis, asthma, rheumatoid arthritis, and cancer. We are mainly focusing on atherosclerosis. So in atherosclerotic patients, it was found that homocysteine is present in very high concentration, but it is still not clear homocysteine is a cause or consequence. In glutathione synthesis pathway, it is found that it is observed. You can see that homocysteine is one of the main precursor of glutathione, and glutathione is homocysteine is converted to cystathine by cystathionine beta synthase enzyme. For that reason, our main target is cystathine beta synthase enzyme. That's why we have treated guinea pig for up to 20 days with cigarette smoke and done HPLC in C18 column in the presence of HBDF, one of fluorescent agent that can bind with thiol group of homocysteine, and found that homocysteine concentration increases increasing. And similarly, we have also found that in cigarette smoke treated Hep cell line and conditioned medium, homocysteine concentration also increases. So from these results, we can see that after cigarette smoke treatment in blood, serum and conditioned medium, homocysteine concentration increases. That's why then we treated macrophage cells with homocysteine and found that after homocysteine treatment and well staining, we have found plaque formation or foam cell formation in macrophage cells. And after that, we have treated CAC treated condition medium in macrophage cell lines and with 6-hour condition medium and 3-hour condition medium, and found foam cell formation after oil staining. Then we have done cystathyon beta synthase enzymatic activity by cell lysers of hepatocyte cell lines in the presence of lead acetate. Homocysteine, when converted to cystathione, it releases hydrogen sulfide. Hydrogen sulfide reacts with lead and given lead sulfide, and we measure the concentration of lead sulfide spectrophotometrically and done enzymatic activity. After doing enzymatic activity, we have found that CBS enzymatic activity decreases significantly after 6 hours. So from these experiments, we can see that cigarette smoke hampers cystathyon beta synthase enzyme activity, and that is one of the potential cause of homocysteine concentration increase. When blood homocysteine concentration increases, it oxidized lipid or low density lipoprotein that is one of the cause of formation of lipid droplet formation. When lipid droplet stores in macrophage cells, and that is the one of the main reason of foam cell formation, and that foam cells build up plaque in the artery, and that is the cause of atherosclerosis. And I want to acknowledge my supervisor, Dr. Alok Kumar Shil, University of Calcutta, for giving support and for financial assistance, Department of Biotechnology, Government of India. Thank you. Is there any...
Thank you, sir. Any questions from the judges?
Yes, Sudipta, how you have
Yes, ma'am.
calculated the enzyme activity?
Ma'am, I have calculated enzymatic activity by per milligram protein, and we have done this enzymatic activity in spectrophotometrically. Per milligram protein per minute, it is converted lead acetate to lead sulfide, and we measured per milligram protein.
Okay. Thank you.
Thank you. Is there...
Thank you. Thank you, sir. Sir, you want to ask any question?
Okay, a small note to Sudipya. Actually, in last slide you have mentioned lipid is oxidized. Basically, there is peroxidation. Thereafter, it leads to the plaque formation. Okay.
Yes, sir.
Okay.
Thank you, Sir Diptya. Next, we have Gulab Chand. You may present now. Gulab Chand, are you there?
Yeah, ma'am. I am audible, ma'am?
Yes, you are audible.
Yeah. Hello to everyone. My presentation title is Development of Lysine and Tryptophan with White Maize Hybrid through Molecular Breeding. I am not able to. Ma'am, there is some problem, I think.
What happened?
One minute. Slide is please, ma'am. Okay. Maize is the staple food of developing country and provide 42.5% world food calories with wheat and rice. Globally, maize consumption pattern is 61% for feed, 17% for food, and 22% industrial and other purposes. By the 2050, demand of maize in the developing country will be double. This is the area and production of maize in world and India. Maize is the third most producing cereal crop. It contributes 9% of Indian cereal food basket and 5% dietary energy supply to the world. And it is the country's tribal population is depend on the maize. And nowadays, population of the India is increasing, and cultivation land are decreasing, and water level of the land are also decreasing. So maize is the perfect crop for the cultivation in India because it required less water and grow in diverse agroclimatic condition. Poverty also play a role in food choice, and maize provides the low-cost food. And this is the maize consumption during the last decade. The consumption is increasing every year, and this is the maize production scenario since 1950 to 51. And this is the maize consumption pattern in India. Around 60% is goes to feed, and 20% is goes to food, and 20% goes to industrial and other purposes. Globally, 820 million are still hungry, and with more than 2 billion people are affected due to insufficient food and nutrition. Nearly 45% of death among the children below 5 are linked with the malnutrition. Among the different undernutrition, higher number of deaths worldwide due to protein energy malnutrition in 2016. It is estimated additional 120 million people could experience protein deficiency at the end of 2050. Kwashiorkor and marasmus are the two major notable diseases developed due to protein and nutrition. It is also affect the economy of the any country. India loses over $12 billion annual GDP due to vitamin and mineral deficiency. Lysine and tryptophan deficiency lead to the fatty growth retardation, lot of hepatitis, and depression and anxiety. This table shows that India is the home of undernourished people. Wasting and stunting are so in the alarming condition. These are the supplementation, commercial fortification, dietary diversity. Biofortification is the strategy to mitigate the malnutrition. Among these strategies, biofortification is the cost-effective and provide a sustainable solution, and it impact on the large population of the any country. $1 investment in the nutritional program offer a benefit of $16. Traditional maize protein is nutritionally poor due to low level of lysine and tryptophan. Discovery of the opaque to lead to the development of QPM, and it have a 2 times higher lysine and tryptophan. Basal and Villegas got the World Food Prize in 2000, and recently Hossain et al. Released 3 QPM variety, Pusa HM4, Pusa HM8, Pusa HM9. But all these variety have only opaque to Z. Opaque 2 alone cannot enhance lysine and tryptophan as required human beings, so we required another gene. And in 2005, Young et al. Mapped a one gene that is called opaque 16 on chromosome 8. Opaque 16 with opaque 2 significantly enhance lysine and tryptophan is maize endosperm. Right now, no white maize hybrid commercialized with both gene, and white maize is preferred as a food and industrial purpose as compared to yellow maize. It is also a good fodder due to digestive property. So keeping over the scenario, we design a work on the biofortification in maize. Material used in the study, we use the parental line of popular white maize hybrid HM5 and HM12. Inbred line are HKI 1344, HKI 1378, HKI 1348-6-2, which is released by the Uchani, and donor parent is developed by Sarika in IRI, and this name is Pusa Q101, which is in yellow color, and this is the phenotype of the recurrent and donor parent. For the interrogation of opaque 2 and opaque 16 gene, we adapt the marker street backcross breeding in this study, and backcross and cell progeny were advanced in 2 places in Hyderabad and Delhi. I start my work in Kharif 2016. We generate a crosses between donor and recurrent parent. After that, we did the back crossing in 2 times, and after that, we did the selfing and developed the inbred line. And this inbred line is based on the phenotypic selection for phenotypic selection, foreground selection, and background selection. In Rabi 2018, we reconstructed the hybrid, and Kharif 2019, we evaluated the hybrids in 2 locations. This image shows the parental polymerase between donor and recurrent parent for OPEC2 and OPEC16 marker. These OPEC2 marker is UMC1066, PHI057. Here, this is clear-cut distinguishes the donor and recurrent parent for OPEC16, UMC1149 and UMC1141 is used, and this is the UMC1149 clearly distinguishes the donor and recurrent parent. For the background selection, we screened more than 500 SSR marker, and we found more than 110 marker, which is show the polymorphism between donor and recurrent parent in each crosses. In foreground selection, we saw the image of UMC1149, which is show the segregation of gene in BC2F2 generation. This is the favorable allele present in this population, and this is the segregation of UMC1066 in BC2F2 generation, and this is our positive plant. On the
Please wrap up in a minute, sir.
On the basis of foreground selection, we did the background selection. This is the recovery of the recurrent parent, and this is the graphical representation of the HKI1348-6-2. There is the recurrent parent, there is the donor parent. Here is the OPEC16 gene was present, and there is OPEC2 gene was, and most of the color is shows is redness. So it is shows the recovery of the progeny. We evaluated the inbred line in IRI during the Kharif 2019. We take the data of dust characterization of inbred and also took the data of yield. Ear and this image show the ear and grain characteristics of mass derived progeny, which is similar to the recurrent parent. We also re-evaluated the inbred line during 2019-20 in Hyderabad, and this is so the coloration in original line, but mass derived line there is no coloration. We also estimate the lysine and tryptophan through UPLC, and mass drive inbred show the 2 to 3-fold higher lysine and tryptophan as compared to original. Mass drive hybrid reconstruct hybrid with OPEC to OPEC16 evaluated at 2 locations in 2 replication possess significance higher lysine and tryptophan. Grain yield potential of the improved hybrid was as par with the original hybrid. This is a peak of ear and grain characteristics of open pollinated improved hybrid. So yield is also good and quality is also improved. So most probably next curve it will be identified for the real commercial release. Conclusion: white maize hybrid assume green significance as a food, cattle feed, and various industrial purpose. Segregation population were successfully genotyped by using marker. We found heterozygous in backcross generation, homozygous in self generation. Background selection lead to the high recovery of the recurrent parent. Selection progeny of OPEC to OPEC16 possess similar plant, ear, and grain characteristics of recurrent parent. Improved hybrid recorded significantly higher nutritional quality and comparable yield to the original hybrid. Thus, mass drive hybrid OPEC to OPEC16 based QPM hybrid hold great potential to elevate the protein energy malnutrition. I want to acknowledge my PhD guide, Dr. Tanu Alan, assistant professor in MIT University. We have to acknowledge the International Center for provide the oxygen crop in field, and also acknowledge the CCS regional station kernel for sharing the inbred line and also piloting the hybrid, and also want to acknowledge for the ICR for funding. And I will especially thank to Dr. Firoz Hussain, principal scientist in IRI, and my external co-guide, and Dr. Vignesh and Dr. Rajkumar also great help as a scientist in IRI and other lab member of our lab. Thanks to all. Thank you.
Thank you, sir. Any questions from the judges?
So apart from the molecular markers,
yes,
have you used any biochemical markers also in the selection process?
No, ma'am. We use the phenotypic selection. We check the similarity and then we decide to generation advance.
You used only SSR markers.
Yes, ma'am. I used only SSR.
So can there be any gene-specific markers which can function?
Yeah. For OPEC2, there is a gene-specific marker UMC1066 and PHI05. For OPEC16, we use linked marker.
Okay.
Yes.
And why you choose lysine and tryptophan
Because
specifically these amino acids? What are their importance?
Yeah, and lysine and tryptophan is essential amino acid, and maize is deficient these two amino acids. That's why we use we use to enhance the lysine and tryptophan. And after the enhancing lysine and tryptophan, it is equal to 90% of the milk. Bioavailability is 90% of the milk.
Okay.
Yes.
So other than these amino acids, any other it's your opinion about the other micronutrient can be enhanced other than this amino acid?
Ma'am,
it can be done?
We didn't estimate the other.
No, it's
amino
just
acid.
a general question, not related to this work.
No, many study.
Any idea?
No, ma'am.
Okay.
Oh, thank you.
Thank you so much. The presentations for the day were really nice. Thanks to all the judges. With this, we now like to conclude the day 2 of ICIBLS. We hope that you had a great day with a lot of new learnings. We thank you all for the enthusiastic participation, and we are excited to see you all for the third day of ICIBLS 2020. Tomorrow, we would be having exciting talks lined up from 10 a.m. IST. We have speakers from different universities, including NIH USA, JNU, and CSIR IGIB. We would be having Dr. Kajal Biswas, Dr. Manjishta Sen Gupta, Dr. Sushil Kumar Jha, Dr. Tulika Prasad, and Dr. Umesh Kumar. Apart from that, we would be also having academia industry interaction with 4 dignitaries from well-known pharmaceutical companies like Cadila Pharmaceuticals, Torrent Pharmaceuticals, Cipla, and Biohouse Solutions to discuss about the academia and industry gap and take up queries about the same. So we would be joining again tomorrow. Hope to see you all there with a lot of enthusiasm. Till then,